this is Mistaken Forward, and I wanted to show you the cell structures that you are going to need to be able to identify. You need to be able to recognize, and part of that is understanding their shape, their location in the cell, their size, any distinguishing characteristics that they have. Now, as we start the cell, one thing that I hope that you really can uh, understand is that the cell is a very dynamic structure. It's alive, and the cell is the basic unit of living things. And so, what we're looking at is a static drawing. It's not moving. It's a diagram. But much like a snapshot of a city, we might see buildings and people frozen in time, but we're not. We understand that that's a picture of a moving entity, a moving city. People move, cars move, buses move. Well, same thing that we're going to see with a cell. This picture just kind of has the cell frozen in time, but in reality, all of these structures are very dynamic. So that's what I hope you're going to gather from what we're gonna study. So we're looking at a eukaryotic cell, and as such, you're going to see a very, very large, centrally located nucleus. And so this would be a cell that's in you. This would be a cell that's in your dog or an amoeba in the pond. Now, this is what we would call a generalized animal cell. Really, what that means is it's not a plant cell. We'll look at that in just a little bit. Notice we have a plasma membrane or a cell membrane surrounding the cell, and that contains it, right? That keeps the boundary. And we'll look at that in a little bit more detail. And so the central parts of a eukaryotic cell would be the plasma membrane surrounding it, and then we have the cytoplasm, all of these organelles that are inside the cytoplasm, and the fluids surrounding the organelles, and then we have this nucleus. So let's talk about that a little bit. The nucleus is going to have three distinct parts to it. The nucleus has the chromatin, which really just means it stains dark under the microscope. It has kind of a grainy or a thready appearance, and what we'll learn is that that's just DNA. These are all the strands of DNA and kind of proteins that are holding the DNA in organized pieces. And we call that the chromatin. Then we have the nucleolus, and the nucleolus is itself just a little darker area. And we'll talk about what its job is, but mainly you just want to be able to recognize where it is. And then the nuclear envelope right here would be the outer layer of the nucleus. It's what surrounds the nucleus. Remember, that's what defines this as a eukaryotic cell. It has a distinct membrane-bound nucleus as well as these membrane-bound organelles. Now, the nuclear envelope also has these little holes in it called nuclear pores. And if you notice, that outer membrane of the nuclear envelope is continuous with this next structure we're going to look at, and that's the endoplasmic reticulum. Now, that's definitely a mouthful, the endoplasmic reticulum. The endoplasmic reticulum is marked by these folds of membrane. Imagine just taking fabric and folding it, folding it, and folding it, and you kind of have the same idea as this rough ER. Now, the rough ER is called rough because it has these little dots, and there are hundreds of thousands of them. I mean, we're talking about upwards of 50 to 100,000 ribosomes in the cell. And some of them are on the rough ER. So the rough ER is called rough because of those ribosomes. But ribosomes can also be in the cytoplasm. You can see where my red dot is. There's a couple of ribosomes right there. And they're in the cytoplasm as well as on the rough ER. So you can see the size difference. Ribosomes are very small. In fact, ribosomes don't have a membrane around them, but they are interacting with the rough ER. Now, the smooth ER is continuous with the rough ER, but it does not have ribosomes on it. Smooth ER also tends to look like little tubes, as not so much the folds. But those are the distinguishing characteristics. Smooth ER does not have ribosomes, rough ER does. If we go a little further out, we see the Golgi body or Golgi apparatus. And you'll notice that the Golgi body, and it's easy to get this mixed up. Here's the key. The Golgi body tends to be a little further out, and it's a little bit more confined to its own space. The rough ER is much more spread out. It kind of surrounds the nucleus. The Golgi body is a little further out, and it's kind of its own body. And it kind of looks like somebody smushed it in the middle. Imagine a stack of pancakes and somebody squeezes it in the middle. And that's the Golgi body. And it tends to have these little circular vesicles that kind of come off of it like a lava lamp. Um, and that's kind of how you would recognize that. Let's see, I think that takes care of over on the right. If we take a look at the bottom, one thing that you'll recognize would be our mitochondria. And the mitochondria are these orange structures. Now, again, this picture kind of shows them down at one side, but they're spread. Here's one over here that's not been cut in half. But the mitochondria kind of have a kidney bean appearance, at least in this picture. And notice they have this intricate membrane on the inside of them. And that's distinctive to mitochondria. They have an outer membrane and an inner membrane. And you can see they're kind of relatively small compared to the rough ER or the nucleus. Coming from this centriole, notice the centriole has these little yellow lines coming from it. And the yellow lines are called microtubules. And the microtubules make up kind of the internal framework of the cell. They create the shape of the cell, they create the structure of the cell. For cells that don't have a cell wall, this is a lot of how the shape of the cell is made. But they can also serve as little transport highways. Very important, these microtubules. The centrioles are kind of centrally located outside the nucleus, and they kind of organize the microtubules. We'll talk about them as well. And then the last thing would be our lysosome. And the lysosomes, they don't look like much. They look like little circles. But they color code them kind of this lavender just to designate that they are different. Lysosomes are kind of hard to label because they do just have an undistinguishing look. They just look like circles. But they're a little unique in what they have. They have enzymes inside of them, and that makes them very powerful. And we'll talk about what they do a little bit later. So those are some of the distinguishing characteristics that you should note. I did forget about the little flagella. I don't have it labeled, but the flagella and the cilia are going to help the cell move or move things past it. Now, plant cells, and this is a plant cell. Let me go ahead and send this to the back so we can see it. So plant cells are going to be distinguished because they have a couple of extra, there we go, a couple of extra organelles that animal cells don't, and then they're missing a couple. So notice plant cells have a chloroplast, which you can see have these little stacks of membranes inside, and they're marked by they kind of look like stacks of pennies. But notice they have an outer and inner membrane, and plant cells have an abundance of them. But notice, and here's something interesting, plant cells do also have mitochondria. That's sometimes the misconceptions people have. You know, we have mitochondria, plants have chloroplasts, but plants, all cells all eukaryotic cells are going to have mitochondria to help provide energy. The other thing the plant cell has is it has a cell wall, which helps govern its shape, and it keeps plants upright and plant cells upright. But they also do have a cell membrane. So you can see right here, they took the cell wall off and it still has a membrane surrounding it. They also have this large organelle that's fluid filled, and this large organelle is a vacuole, and that's what it's usually filled with water. Sometimes it stores nutrients as well. Uh, so see if you can recognize some of these other organelles, and then you can put them in their place. Some of these we can do together. And so before I leave you, we can do some of them in my video. So here you have the Golgi body. Here you'd have the nucleus. The mitochondria would be our orange, and you can put lines in if you'd like. Smooth ER is kind of tucked right next to the rough ER, hard to see. And then the rest of them, you can't really, it's hard to label with this one because they're so jam-packed with the vacuole. So let me go ahead and put the vacuole in just so, there you go. So that is your introduction to the cell parts. And you can practice labeling yours on your Jamboard. Have a nice day.